Good day, everyone. Our topic for today is all about the electron transport chain and oxidative phosphorylation. Next. The electron transport chain, or ETC, facilitates the passage of electrons trapped in FADH2 and NADH during citric cycle. So, ang FADH2 and NADH contains electrons which are contained to this set of enzymes and complexes. ETC is the series of biochemical reaction in which intermediate carriers, protein and non-protein, aid the transfer of electrons and hydrogen ions from NADH and FADH2. Next, the ultimate receiver of electrons is molecular oxygen. The electron transport or respiratory chain gets its name from the fact that electrons are transported to oxygen absorbed via respiration. ETC is the sequence of reaction whereby the reduced forms of the coenzymes are oxidized ultimately by O2. The enzymes and electrons carriers indeed for the ETC are located along inner mitochondrial membrane. They are organized into four distinct protein complexes and two mobile carriers. Next. These are complexes are numbered from 1 to 2, and we also have additional prosthetic groups called the coenzymes Q and cytochrome C. So the first, let's talk about complex number 1. Complex number 1 of the electron transport chain is named as NADH dehydrogenase complex, and it also known as the NADH oxyureductase. So the complex number 2 is called succinate dehydrogenase complex because it removes hydrogen from the compounds, and in this case it will remove hydrogen from the succinate and oxidize into fumarate. So complex number 3 is called the cytochrome reductase, and it also known as the Q cytochrome C oxidase. So lastly, the complex 4 is called the cytochrome C oxidase, and it is a copper-containing complex whose main job is to oxidize cytochrome C that is receive electrons from cytochrome C, and then it uses those electrons for the reduction to water. Okay, next.
So,
Okay.
so in the complex one, as stated earlier, it is also known as the NADH ubiquinone oxidoreductase. It is made up of NADH dehydrogenase, flavin mononucleotide or FMN, and 8 iron sulfur clusters. So, NADH dehydrogenase here is an enzyme. When we say enzyme, it is a substance that acts as a catalyst in living organisms that converts nicotinamide adenine dinucleotide or NAD. So NAD is a coenzyme central to metabolism. So the flavin mononucleotide or FMN here is a biomolecule produced from riboflavin and the 8 iron sulfur clusters as cofactors. So next, let's see the process of the complex 1. So the NADH or the nicotinamide adenine dinucleotide and hydrogen donated from glycolysis, and the citric acid cycle is oxidized here, transferring two electrons from nicotinamide adenine dinucleotide and hydrogen to flavin mononucleotide. Then they are transferred to the 8 iron sulfur clusters and finally from 8 iron sulfur clusters to coenzyme Q. During this process, four hydrogen ions passed from the mitochondrial matrix to the intermembrane space, contributing to the electrochemical gradient. So the intermembrane space is a compartment of every small volume since the outer and inner membranes are in very close proximity. So complex 1 may also play an important role in causing apoptosis and programmed cell death. So balik ta sa last slide. Next is complex 2. Complex 2 is also known as succinate dehydrogenase. It accepts electrons from succinate. So succinate is an intermediate in the citric acid cycle. Acts as a second entry point to the electron transport chain or ETC. When succinate oxidizes to fumarate, so next nata sa, let's see the process of the complex 2. So when succinate oxidizes to fumarate, two electrons are accepted by flavin adenine dinucleotide or FAD within complex 2. Flavin adenine dinucleotide passes them to iron sulfur clusters and then to coenzyme Q, similar to complex 1 or similar to NADH ubiquinone oxidoreductase. However, no protons are no protons are translocated across the membrane by complex 2. Therefore, less adenosine triphosphate or ATP is produced with this pathway. So next here is this is the overview of the complex 2. So the FADH here is produced when acyl CoA is oxidized to enoyl CoA in the first step. And last, coenzyme Q, also known as ubiquinone CoQ, is made up of quinone and a hydrophobic tail. Its purpose is to function as an electron carrier and transfer electrons to complex 3. Coenzyme Q undergoes reduction to semiquinone and ubiquinol through the Q cycle. This process receives further elaboration under complex 3. So,
So let's proceed to complex 3, which is the coenzyme Q cytochrome C reductase. So several iron sulfur proteins and cytochromes are electron carriers in this complex. Cytochrome is the hem iron protein in which reversible oxidation of an iron atom occurs. So complex 3 is also known as the Q cytochrome C oxyreductase or cytochrome reductase. So in this process, we there are several important structures that we encounter in this process. First is the cytochrome C1, which contains one hem group. Next is the cytochrome B, which contains two hem group. And next is the rescue center or 2Fe, 2S group, 2S group. So more to elaborate this process more, I downloaded another process or photo about this process to explain well. So this process by which electrons travel the QH2 to cytochrome C is known as the Q cycle. So this step 1 and step 2 is known as the Q cycle. This cycle begins when QH2 binds to the complex 3 upon complex 3. Upon binding, the two elements electrons follow different paths. So these are the paths that the electrons follow. One electrons move to 2Fe2S group of the risky center and then transferred onto the heme group of cytochrome C1. So the first electron moves here. Then oh my god. It is then picked up by cytochrome C, which diffuses away and travels to complex 4. So the first electron travels here, then goes out something there. Then the second electron moves into the hem groups of cytochrome B before being picked up by ubiquinone Q or coenzyme Q. It is then reduced to form semiquinone radical ion Q plus. So in the next step, it is still the same. Then these two steps are combined together to form or to produce or release 4H plus. So in conclusion, in one Q cycle, there are two QH2 which are oxidized into Q, releasing 4H plus. One Q is reduced into QH2. Then two cytochrome C molecules are reduced. So next is the complex 4. Oh, Jesus. Complex 4 coenzyme Q cytochrome C reductase. The electrons flow from cytochrome C to cytochrome A to cytochrome A3. In the final stage of the electron transfer, the electrons from cytochrome A3 and hydrogen ion plus combine with oxygen to form water. So in this process is very simple and understandable. So in cytochrome C, there are 4 electrons here. Then it will be transferred to copper A. Then the electrons from copper A will be transferred to the cytochrome A. Then the electrons from cytochrome A then transferred to the copper B. 4 electrons from cytochrome C are required to totally reduce O2 to water. So in this final stage of electrons transfer, the electrons from cytochrome A3 and hydrogen ion combine with oxygen to form water. So, mana to siya. Next. OMG. So, in the summary of flow of electrons through 4 complexes of the electron transport chain. So, these are the summary of the process. In complex 1, it facilitates transfer of electrons from NADH to coenzyme Q. In complex 2, it generates FADH2 and generates COQ as the final recipient of electrons from FADH2, which will be used in complex 3. In complex 3, it accepts electrons from ubiquinol and transforms them into another electron carrier called cytochrome C, which is then used in complex 4. So lastly, in complex 4, it functions to transfer electrons from cytochrome C to oxygen and the process to form water and help generate a proton gradient. So that's all for the electron transport chain. Let's proceed to the oxidative phosphorylation.
So oxidative phosphorylation. Oxidative phosphorylation is a closely linked process to the electron transport chain, which is through by which is produ- pro- processed by coupled reactions. So oxidative phosphorylation also is the catalyst in which uh, ATP or adenosine triphosphate is synthesized from adenosine diphosphate and PI or inorganic phosphates using the energy released in the electron transport chain, but of course the coupled reactions. So what are coupled reactions? Coupled reactions are pairs of biochemical reactions that occur concurrently. So when we say concurrently, it occurs at the same time. The biochemical reactions occur at the same time in which they release energy that is used by the other reaction from the first reaction. So an example of coupled reactions is the oxidative phosphorylation and the oxidation reactions of the electron transport chain are coupled systems or coupled reactions. So next, the coupling of ATP synthesis with the reactions of the electron transport chain or ATC is related to the movement of protons or H plus ions across the inner mitochondrial membrane. So the process of the movement of the protons is typically starts from the matrix towards the inner mitochondrial membrane and outside towards the intermembrane space. So as as it's established before, oxidative phosphorylation and the electron transport chain is closely linked. So this means that the complexes of the electron transport chain also plays a huge role in the oxidative phosphorylation process. So more specifically, the complexes 1, 3, and 4 of the electron transport chain They serve as the proton pumps in which they transfer protons from the matrix side of the inner membrane to the intermembrane space, and then I matrix side of the inner membrane or the inner mitochondrial membrane and outside to the intermembrane space. So for every two electrons pass through the electron transport chain, four protons cross the inner mitochondrial membrane through complex 1, and four through complex 3, and two more through complex 4. So next. This proton flow causes a buildup of H plus or protons in the intermembrane space. So the high concentration of these H plus ions or protons in the intermembrane space becomes the basis for the ATP synthesis. So when you so let us focus our attention towards the diagram. Here we can see the intermembrane space, the inner membrane or the inner mitochondrial membrane, and the matrix. And inside the matrix of the mitochondria, diri nag-occur ang electron transport chain. So then we can see the complex 1, complex 2, complex 3, and complex 4. So sa complex 1, 4 H plus ions pass through is transferred from the matrix, which then passes to the inner mitochondrial membrane and outside into the intermembrane space. So the same can be said to the complex 3. Four, four, 4H plus ions are also released from the matrix towards the inner membrane and outside the intermembrane space. Also, in complex 4, 2H plus ions are instead from the matrix towards the inner membrane and outside the intermembrane space. So next.
So the resulting concentration difference high in intermembrane space than in matrix constitutes an electronic chemical proton gradient which is always associated with potential energy. The gradient build-up would spontaneously push the H plus ions through membrane-bound ATP synthase. Proton flow is not through the membrane itself. It is not permeable to H plus ion. The proton flow through the ATP synthesis power the synthesis of ATP. ATP synthesis are coupling factor in the ATP or OP coupled reaction. So the hydrogen ions or H plus in the matrix space can only pass through inner mitochondrial membrane through a membrane protein which is ATP synthesis. As protons, it moves ATP synthesis. The ADP turned into ATP. The production in ATP using the process of chemosynthesis in mitochondria called oxidative phosphorylation. So next. So in this process, in which the energy is from protein gradient, which used to make ATP, in what we call chemosynthesis. So the chemosynthesis can refer whatever process and energy stored in a protein gradient in which used to work. Although chemosynthesis accounts for over 80% of ATP made during glucose breakdown in cellular respiration. It's not unique for cellular respiration. The chemosynthesis is also involved in light reaction to photosynthesis. The continuation of the topic will be discussed by the group 6B.
That's all for our group. Thank you for listening. Bye bye.